சரி கொஞ்சம் நவுந்துக்கலாமா? கரெக்டா தெரியுதா? இல்லை இப்படி கொஞ்சம் நவுந்துக்கலாம். டிவியோட ப்ராப்ளம் இப்படி இருந்தலாம் இந்த பக்கம் ஒயிட் கார்டு சரி இன்னொன்று இல்லை இல்லை ஓ லாஸ்ட் வீக் நீங்க இல்லாம ரொம்ப கஷ்டமா இருந்துச்சு. ஷேர் பண்ணிடலாம் இல்லையே இருக்கு. ஒரு தூங்கிட்டு என்ன எழுப்பி விட்டுருங்க. வேற ரொம்ப அப்படியே சொக்குது கண். வேண்டாம். சூப்பர். ஓ அங்க பாட்டில் எல்லாம் பாத்தீங்கன்னா பூஸ்ட் ஏதோ இருந்துச்சு. நம்ம சாப்பிடுறதுக்கு தானா? பரவாயில்லையே. ரெண்டு மைக் ஆன்ல இருக்கு.
Namaste, sir.
Hello, ma'am. Good evening, ma'am. Can you hear me?
I said it's not a correct time. Please move forward. Turn this way. Turn this way.
Mean fan கூட ஆஃப் பண்ணிடுங்க சார். Hi students, good evening. Hi students, good evening. Can you all able to hear me? Yeah, good. Am I audible to you all?
Yes, sir.
Yes, ma'am. Good.
Move forward, maybe. Charts, be there, and the next question. Both are partakers of the discussion. And none of them are.
Oh, that's for max classes. That's for max. So, dear students, can you all able to see the screen which I shared? Ma'am, my screen is visible to you all. I'm just sharing
No,
my.
sir.
Ma'am,
Sir, it is not visible, sir.
not visible. Share பண்ணிருக்கேனா? Oh. நீங்க ஃபர்ஸ்ட் பண்ணீங்க தானே சார்? And just check now.
Yes, sir, it is visible, sir.
Okay. Shall we start? Hello students, are you all ready? Shall we start?
Yes, sir.
Yes. Okay. In the last class, we discussed about primarily about what is photosynthesis, and then the photosynthetic pigments, and then the absorption maxima of the photosynthetic pigments. Okay, so primarily about the photosynthetic pigments we saw. Okay, now in today's class, we are going to discuss about the mechanism of photosynthesis, and then the Z scheme, the Z scheme of photosynthesis, about the photosystems we are going to study, and about the difference between the dark and light reactions we are going to study. Okay, and then the cyclic, non-cyclic photophosphorylation. If possible, we study both of the things. And then the chemi-osmotic synthesis. Okay, so only this part, very little part, we are going to discuss today. Okay, I just show you what and all we are going to discuss, just for you to have an idea, like the mechanism of photosynthesis. Okay, and then the difference between the light and dark reactions, and then what occurs in the light reaction, and the important process connected with the light reaction, the photolysis, or once again, Bhaskar sir, just a minute. Okay, so photolysis and then the pigment systems, their functions basically, the pigment systems, and then like the photosystem 1 and photosystem 2. Okay, hello students, can you hear me? Yeah. Okay, fine. Anyway, so sorry for the inconvenience, some technical issues. Now, fine. So let's start. First, coming to the mechanism. Hello, ma'am, is it okay or any other issues still there?
Nothing, sir. No issue, sir. You can let us begin, sir.
Okay, thank you. So, mechanism of photosynthesis. Okay, so photosynthesis is an oxidation reduction process. Sometimes they may ask a need. Okay, so it is an oxidation reduction process. Okay, oxidation reduction process. And then water is oxidized. So look at that. Water is oxidized to release what? O2. Okay, O2. And CO2 is reduced. Okay, so oxidation of water and reduction of CO2. CO2 is reduced to form starch. Okay, water is oxidized. Sometimes they may ask, what is oxidized? What is formed? So water, the H2O is oxidized and to release what? O2, oxygen. And CO2 is reduced to form what? Starch. Or the simple sugars. Simple sugars are formed and then they are converted into complex polysaccharides. Okay, fine, good. So this oxidation and reduction process two phases, or the photosynthesis occurs in two phases, as everybody knows. So, fantastically, the two processes are named, like everybody can remember the phases of the process. First phase is the light phase, or you can say light reaction, light phase, light reaction. And also this is called what? Light dependent reaction. You can say Hill's reaction. Okay, this is Hill's reaction. So light phase, dark phase are the two phases of photosynthesis. Okay, first phase is light phase and then dark phase. And first one is light dependent. Okay, directly connected with light. And second phase is actually, though they call it as dark phase, it does not mean that it occurs in dark. Okay, it does not mean that it occurs in night time. Means it does not require light. Okay, and it is the continuation of light process. Okay, it is the continuation of light process. But fascinatingly, they named it as light phase and dark phase. Okay, so the other name of light phase is Hill's reaction. So that is name why it is called Hill's reaction, based upon the name of the scientist. Okay, Hill's reaction, name based on his name, it is called what? Hill's reaction, Robin Hill's reaction. Okay, what occurs in simple? What occurs in this light phase? Look at that. Energy from sunlight. Okay, during this phase, energy from sunlight is absorbed. Okay, energy is absorbed. And the sunlight is actually in what form? Physical form. It is there in what form actually? The physical form. Okay, it is there in the physical form. And the physical form of energy is converted into chemical form. That's actually photosynthesis. But what is produced? The main thing which you should understand here is, what is the product of light reaction? Look at that. You can say ATP is produced here, and then NADPH, H plus. So the products what are formed in light reaction are very important. They, you can say either NADPH plus H plus or NADPH2. Okay, NADPH2. Okay, so they are the products of light reaction. And then dark reaction. In photosynthesis, the dark reaction is actually light independent reaction. It is light independent. Earlier one is light dependent reaction. So this is light independent reaction. Okay. See, they call it as Hill reaction. Light reaction is also called what? Hill reaction. So the dark reaction is also called what? Blackman reaction. Okay, it is called what? The name of the scientists are very important. Blackman reaction. Innate point of view, you should know Blackman reaction and Hill reaction. Light reaction, dark reaction. Okay, fine. So this is biosynthesis phase. Also, this is called what? Biosynthesis phase. And then here, what? Look at that, carbon dioxide fixation occurs. Okay, the carbohydrates are synthesized. That's the very important thing. Okay, so in the light reaction, the products are primarily ATP and NADPH2, and then here in the dark reaction, carbohydrates are synthesized. Okay, the carbohydrates are synthesized. Okay, and ATP, NADPH2 are utilized actually. They are utilized. Okay, and then carbon dioxide means carbohydrates are produced. So keep this idea in mind. What you should know, you know, maybe generally what is light reaction, what is dark reaction, but you should know the other names. Okay, Hill's reaction, Blackman reaction, the products ATP, NADPH2 in light reaction. In dark reaction, it is biosynthesis phase. Carbohydrates are formed. CO2 fixation occur. Okay, so keep that in mind. Now, this is also very important. look at the difference between light reaction and dark reaction. Light dependent process. Okay, light reaction is light dependent process. And here it is dark reaction does not require light. Light dependent reaction, and here dark reaction does not require light. And then here, photosystem 1 and photosystem 2. We are going to study about this. They call it as what? PS1 and PS2. They call it as what? PS1 and PS2. Photosystem 1 and photosystem 2. They are involved in light reaction. But in dark reaction, no photosystem required. Okay, photosystem, because it does not require light. So no photosystem required. Okay, fine. And the third important aspect of light reaction is photolysis. See, look at it. Photolysis of water means water is split. Splitting of water. Photolysis is nothing but splitting of water. H2O is water. That splitting of water is occurred. So, look at it. Oxygen is liberated. Okay, oxygen is liberated. So, product of photosynthesis, oxygen is liberated because of what? Hydrolysis of or photolysis of water. Okay, photolysis of water. And then here, no photolysis. Okay, does not take place. Photolysis does not take place. But here, oxygen liberated, CO2 absorbed. Look at the difference. In light reaction, oxygen is released, liberated. But in dark reaction, CO2 fixation, carbon dioxide is absorbed. The difference between light and dark reaction. And then the location where it occurs. Look at that. Light reaction occurs in the grana. Okay, grana of the chloroplast. Of course, in chloroplast, so grana of the chloroplast. The dark reaction occurs in the stroma. Okay, stroma of the chloroplast. Okay, grana of the chloroplast. Light reaction occurs. Stroma of the chloroplast. Dark reaction occurs. Okay, so that's very, very important. And then look at the products. ATP and NADPH. They are actually the products. Okay, of dark reaction. Here, glucose is the product of light, sorry, ATP and NADPH are the product of light reaction, and glucose, CO2 fixation occurs, and glucose is the product of dark reaction, and glucose is converted into polysaccharide. Okay, carbohydrate. So, difference between light reaction and dark reaction. Okay, so it's very, very important. And then, so now let's enter into like important aspect of the light reaction. What are the important aspects, important thing we must know about the light reaction? Okay, so about the light reaction. Okay, so look at the other names. Light phase, photochemical reaction, light dependent reaction, or Hill reactions. All are same only. Light phase, dark phase. So light phase, photochemical reaction, and then light dependent reaction are also called Hill reaction. Okay, fine. Based on the scientist Robin Hill, you can see in the second paragraph. Okay, fine, good. As we said, light reaction occur in grana, and then which includes what? Assimilatory powers. Okay, here, of course, the rest of the thing you should know, dependent on light. And then, so assimilatory powers like ATP and NADPH2. Okay, ATP and NADPH2 are the products produced in light reaction. Okay, fine. So, Robin Hill, remember the scientist name? Robin Hill only first showed chloroplast extracted from leaves, and sometimes they may ask the name of the plants also. Stellaria media and Lamune album. Basically, they are suspended in a test tube containing electron acceptor. Look at that. Sometimes they may ask about this electron acceptors also. Note down the name of the chemical, potassium ferroxylate. Okay, potassium ferroxylate. Okay, so and potassium ferricyanide, potassium ferroxylate, and then potassium ferricyanide. Okay, look at the first thing. When chloroplast, okay, Robin Hill demonstrated that if chloroplast extracted from leaves, okay, leaves of certain plants, and then that chloroplast is suspended in solution, the suspension should contain, look at the chemicals, potassium ferroxylate, potassium ferricyanide. Name of the chemical is very important. So that time oxygen is released due to splitting of water. Okay, oxygen is released due to splitting of water. So you need what kind of questions they may ask in Hill's reaction? What chemical substance was used as electron acceptor? Okay, they may ask in Hill's reaction or in light reaction, what chemical substances are used as electron acceptor? So you should write about what electron acceptor? Potassium ferricyanide or potassium ferroxylate. Okay, so they are the chemicals which used as an electron acceptor. Okay, fine. And then what is released here? What is released here in light reaction? Oxygen is released. That's very, very important. Okay, before the utilization of CO2, you just know about this. Oxygen is released. Okay, oxygen is released. And already in the previous slide, we saw oxygen is released because of what? Photolysis. Okay, photolysis. Okay, of water, splitting of water, splitting of H2O. Because of that only oxygen is released. Okay, so under these conditions, look at that, no CO2 consumed. In light reaction, what is not consumed? No CO2 was consumed and no carbon dioxide, carbohydrates are produced. No CO2 released, no carbon dioxide produced. Okay, and then look at that chemical reactions 4Fe3 plus. Actually, these chemicals with the functional group act as electron acceptor, and then the electron acceptor is, for example, in this experiment, potassium ferricyanide. What is the electron donor? See here, splitting of water. Okay, that splitting of water releases oxygen as well as provides electron for the reaction. Okay, so that's very, very important thing. And then now 4Fe3 plus reduced to 4Fe2 plus. 4Fe3 plus reduced to 4Fe2 plus. And then look at that, this part. So 4H plus are liberated. Okay, 4H plus are liberated. And then oxygen. Oxygen molecule is actually released. So they will ask this for what? Splitting of water. So this is how, okay, the splitting of water occurs. Okay, splitting of water. That chemical reaction you have to remember. Okay, next. So same thing, let us discuss a little more in detail about with some experimental proof given by other scientists. See, this chapter really, like, you need to understand some kuti things, very simple things. See, it's much about facts and figures. How well you all remember the facts and figures helps you to understand, and then you can able to answer the questions. Okay, fine. Now, photolysis, splitting of water. Photolysis is nothing but splitting of water. Photolysis is splitting of water. Okay, that's the first important step. And this is known as what? Hill's reaction, as we said. Okay, so first step, look at that, the reaction. Hill's reaction, what it says. First, oxygen is released from water. Splitting of water occurs, so oxygen is released. And then the second important thing is the electrons from for the reduction of CO2 are obtained from water. So this is the thing which we saw. Okay, electrons and then splitting of water. Okay, so that's the important thing you should know. Okay, and then another important thing is actually look at that. So they use in Hill's reaction to prove the mechanism of photosynthesis, they use a dye. Sometimes they ask the name of the dye. At least remember the short form, abbreviation of the dye, DCPIP, DCPIP. What is DCPIP? 2,6 dichlorophenol indophenol. 2,6 dichlorophenol indophenol. Okay, so this is the dye actually used in Hill's reaction. Okay, so this color also sometimes known as blue color chemical substance uses redox dye. And to prove this, yes, water is released, means water is splitted and then O2 is released and iron is released. So to understand that this chemical reaction basically dye test, they call it as also dye test. It helps basically. Okay, fine. So when these chemicals exposed to light, and it represents how the photosynthesis initially occurs. And then and Hill's reaction. So remember this Hill's reaction and then R known reaction. Okay, R known experiments. Here it proves that the light energy is converted into chemical energy. Okay, so this experiment importance is here it talks about splitting of water. Hill's reaction talks about splitting of water, and here it talks about how the light energy is converted into chemical energy, the mechanism of conversion of light energy into chemical energy. So what is called the chemical energy? ATP. ATP is called what? The energy currency of the cell. ATP. Okay, how ATP is formed. Okay, so the energy currency of the cell. So that's very, very important. So our known experiment. Okay, fine. And then here, look at that electron acceptor R. Okay, in Hill reaction, what was the electron acceptor? Anybody can tell me? Potassium ferricyanide, potassium ferroxylate. They are the electron acceptor. But here in iron reaction, they may ask electron acceptor, basically NADP plus. Okay, so NADP plus. That's the electron acceptor. Okay, fine. Next important thing. So look at that. So in photophosphorylation, here in Arnold reaction, NADPH is formed as hydrogen donor. Okay, NADPH is formed as hydrogen donor. So electron acceptor and then hydrogen donor. Okay, what is the electron acceptor? NADP plus. What is the hydrogen donor? NADPH. Okay, that's the hydrogen donor. So these little thing you should remember, actually keep remembering those things. Okay, so these two molecules, formation of hydrogen donor. Okay, so this is the hydrogen donor and electron acceptor. Okay, is known as photo reduction or production of NADH. Okay, so here photolysis, first reaction is photolysis and then photo reduction reaction. So both are important in photosynthesis, photolysis and photo reduction. See these things, when you can well connect this, actually when you come across some questions related to photosynthesis, this part, these names and these chemical substances are very, very important. Okay, now fine. Now, so the pigment system. So as of now, we should understand about splitting of water, like Hill's reaction, okay, and then this Arnold reaction, Hill's reaction and Arnold reaction, okay, fine. And what kind of, whether it is oxidation or reduction, so that type also you should understand. Now, so pigment systems, okay, so light LHC, know about this term? Two discrete photochemical light harvesting complexes. There are two types, like light harvesting complexes. Okay, light harvesting complexes. And then PS1, photosystem 1 and photosystem 2. Okay, photosystem 1 and photosystem 2. Okay, so they too play a crucial role in photosynthesis. Okay, so because we know about one thing called ETC. ETC stands for electron transport chain. Okay, ETC stands for electron transport chain. So this chain actually primarily comprises of this photosystem 1 and photosystem 2. Okay, fine. So what forms this photosystem? What forms the photosystem? You can see a single chlorophyll molecule. Okay, a single chlorophyll molecule in the center. You can see a single chlorophyll molecule in the center, which forms the reaction center. Okay, and then look at that. There are many other substances, some 250 to 400 other pigments surrounds this. See, like this. One is the chlorophyll A forms the center, and all the pigments, other pigment substances or pigments involved in photosynthesis surrounds this. They actually forms this complex. Okay, once chlorophyll A in the center, all other pigments in the peripheral proximity. Okay, so that's very, very important. Okay, and please understand the difference between like this photosystem 1 and photosystem 2. Okay, fine. So chlorophyll A, and then look at the absorption maxima. Okay, these values are also very, very important. Okay, at what particular wavelength they absorb light. Okay, photosystem 1, at what particular wavelength it absorbs light. So that's very important. And then photosystem 2, at what particular wavelength it absorbs light. See, we saw in the last class the WIP-GR, one reaction basically. Okay, the white light is passed to the prism, and then there are spectrum of colors, and photosynthesis occurs in two regions. We saw blue and red region. Within that only, the majority of photosynthesis occurs blue and red region. For example, 400 to 600 700 nanometer. Okay, fine. So that part, please remember here also. Okay, fine. Now, main pigment is chlorophyll A. What are the other pigments? Okay, in photosystem 2, you have other different pigments also. Chlorophyll B and other substances are also present. Okay, fine. So this looks theoretically important thing, but now if you compare it with the table, okay, you can easily understand what is photosystem 1 and its significance, and photosystem 2 and its significance. Okay, see, look at that. Even before getting into that, two important process you can understand. Like something is called what? They ask you what is resonance transfer, and then inductive resonance. Okay, these two important things you should know: resonance transfer and inductive resonance. See, all these things are what today we are discussing as of now, or all are very technical terms. Okay, so please remember things. Okay, fine. Just சும்மா கேட்டு வச்சுக்கோங்க. பின்னாடி when you study about photosynthesis of that phases and all, these terms will definitely help you. Okay, just hear about these terms and keep that process in mind. Okay, fine. Basically, what we are doing now, these are the prerequisite to understand what photosynthesis is. If you understand all these things, then easily you can understand what light reaction, what dark reaction, how the cyclic, non-cyclic photophosphorylation, all everything occurs. Okay, fine, good. So now, the antenna or accessory pigments. Look at the process. Antenna with that accessory pigments receive radiant energy and transfer it. Okay, they receive and transfer. They receive and transfer radiant energy. So, the process by which accessory pigments receives and transmits or transfers radiation energy among themselves is called what? Resonance transfer. Okay, resonance transfer. And then the antenna only receives this resonance transfer, and then these molecules, after receiving the energy, they are excited. the molecules are excited and then they transfer their energy to chlorophyll A molecule. So, accessory pigments only receives the radiation or energy. Okay, accessory pigments receives the energy and then they transfer it to what? Chlorophyll A. This is very, very important in one more point of view. So, chlorophyll multiple choice questions this is very important. So, chlorophyll A first by accessory pigment, then by chlorophyll A in the reaction center. So this flow from accessory pigment to chlorophyll A, the reaction center, this flow, they actually call it as what? Inductive resonance. Okay, this flow, inductive resonance. Okay, and then finally what occurs? This chlorophyll A molecule only converts that light energy into electrical energy. Okay, by bringing electric charge separation. Okay, so this is the chlorophyll A then converts that light energy into electrical energy. Light energy into electrical energy. Okay, fine. Now, look at the difference between the photosystem 1 and photosystem 2. So, earlier we discussed about two important things, light reaction, dark reaction. So, the differences we saw. And now here, please understand photosystem 1 and photosystem 2. Because in electron transport chain, this understanding is very, very important. Okay. Photosystem 1 lies on outer surface of thylakoid membranes. Okay. So you know the thylakoid membranes and in the outer surface only the photosystem on. Okay. Photosystem on is present. And photosystem 2, look at that inner surface, outer surface of thylakoid membrane, thylakoids, and here inner photosystem 2 is inner surface of thylakoids. Okay, photosystem 1 and photosystem 2. And another one is, look at that, in photosystem 1, oxygen is not evolved. Okay, in photosystem 1, oxygen is not evolved. But here, oxygen is evolved because here no photolysis, no photolysis, but here photolysis occurs. Okay, photolysis occurs. And then the reaction center is P700. For photosystem 1, the reaction center is actually P700. It is P680. This is very, very important thing. Okay, P700, P680. And then here NADPH is formed in photosystem 1. Here no formation of NADH plus. Okay, this comparative study is very, very important. Okay, outer surface of thylakoid, inner surface of thylakoid. Oxygen is not evolved, oxygen is evolved. Reaction center is P700 and here it is P800. NADPH formed, NADH not formed. And then participates in, okay, so this photosystem 1 participates in both cyclic and non-cyclic photophosphorylation. Photophosphorylation with the help of light, phosphate group is added. So photophosphorylation. Okay, and here the photosystem 2 participates only in non-cyclic photophosphorylation. See, photosystem 1 participates in both of the cyclic and non-cyclic photophosphorylation. And here it participates only in non-cyclic photophosphorylation. Okay, and then here it receives electron from photosystem 1, receives electrons from photosystem 2, and photosystem 2 electrons receive electrons from photolytic dissociation of water. Okay, by water dissociation, and then not related to photolysis of water, and here it is related to photolysis of water. So keep remembering these things about the basic thing about photosystem 1 and 2. And now, so I think hereafter it is very easy for you to understand. So they are basically the technical terms, technical thing. Now, electron transport chain. As you know, cyclic photophosphorylation and non-cyclic photophosphorylation. Okay, fine. So, in cyclic photophosphorylation, only PS1. See, first, we are going to, that's the simple part. That's why before getting into Z scheme, we are first starting with cyclic photophosphorylation. Then we will go to both of the thing. Okay, fine. So, for simple thing, photo system 1 and photophosphorylation. Okay, so here look at that electron transport chain. PS1 is activated by light order wavelength 680 nanometer. Grano of thylakoid as we discussed earlier. And PS1 is the reaction center. ET transport chain ejected from the reaction center of PS1. Here, in cyclic electron transport chain, no oxygen is evolved. Another important thing, no photolysis, no oxygen. So that's what we discussed in that comparison table. And here, but very, very important thing, what is formed in cyclic photophosphorylation? Okay, like see what products are formed is very important basically. Okay, see ATP. Look at that. Phosphorylation takes place at two places. Thus, in cyclic photophosphorylation, look at that. Two ATP molecules are generated in each cycle. This number is very important. Two ATP molecule. Okay, two ATP generate in each cyclic electron transport chain. And another important thing, NADPH2 is not formed in cyclic process. NADH2 not formed in cyclic phosphorylations. Okay, and the molecule plastocyanin is actually like a copper containing blue protein involved in this process basically. Okay, and then the electron acceptor is actually ferridoxin. Okay, ferridoxin. Okay, this molecule name is very, very important. Act as electron acceptor. Okay, so now look at the cycle. See, in one part, cyclic photophosphorylation. Okay, so photosystem 1. This is the thing which you technically, to make you understand this reaction better, I just gave a lot of explanation. Separately, we discussed about this one, and separately we discussed about this one. And also we discussed about electron acceptor molecule in each of the reactions. Now look at that. Light. The radiation. What radiation they call? PAR. PAR actually stands for what? Photosynthetically active radiation. Okay, photosynthetically active radiation. So that radiation, when it reaches the photosystem 1, okay, look at that, the wavelength, the absorption maxima here, 700 nanometer wavelength. Okay, high, red region. You can say that red region, light. So P700, photosystem 1, photosystem, that 700, P700, and the light is like when the photosystem 1 reaches, means the light reaches the photosystem 1, you can see excitation occurs, and then electron forms, electron transported, and then electron acceptors, chemical molecules are there, they accept electrons, excited normal state to excited state, and then again look at that, the ETC, the electron transport chain system. Okay, so here, because of this pathway, look at that, this is continued again and again. So that's a very, very important. You can see the arrow mark. Okay, you can see that basically. Yeah, see, look at that. This one, in cyclic photophosphorylation, you can see ATP. Look at that. Because of this electron transport chain, because of the photosystem 1, ADP to ADP, the phosphorylation occurs. Again, in one cycle, they said 2 molecules. In one cycle, 2 molecules. So again and again and again. Okay, it is repeated. What only is formed? ATP is only formed. The product of light reaction, ATP and NADPH2. Of course, oxygen is also the product of light reaction. Remember that oxygen is also, but here it is for the biosynthetic phase, they are going to be used. So ATP again is formed. So this is cyclic phosphorylation. Okay, and next, the Z scheme, or you can say non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation. So please, let's directly enter into this electron transport chain. Okay, this electron transport chain. Look at that. So this is very, very important. Okay, in your textbook and all, they first start explaining this only. Okay, they first explain the Z scheme of photosynthesis. Okay, that light reaction, Z scheme. Okay, so this is very, very important. So please understand here. Photosystem too. And the common thing, like as a layman science, what you should understand about photosynthesis is first photosystem 2 comes and then photosystem 1 comes. This 1 and 2 is not the order in which electron transport chain occurs. It does not represent photosystem 1, photosystem 2 is not the representing the order of the reaction, but it represents the order of discovery when they discovered what. At first, photosystem 1 was discovered and then only photosystem 2 was discovered and the mechanisms we understood. But in electron transport chain, in the Z scheme, okay, photosystem 2 only comes first and then only photosystem 1 comes. So this diagram is very, very important. But here, what are the electron acceptor, electron donor, what are the other molecules involved? You should know. Now let us directly enter into this. See, look at that. Light reaches the light harvesting complex. LHC, already we explained. Light harvesting complex. One central chlorophyll A, and surrounding the central chlorophyll A, there are many 200 to 250 to 400 accessory pigments are there. Okay, so the accessory pigments receives the light and then they transfer the light into what? The reaction center, the chlorophyll A. That is one thing. Look at that here. This is very, very important. What this process? Can you understand? This process is actually called what? That splitting of water. So this is called what? The splitting in Hill's reaction, in light reactions. Before that, we saw photolysis. So this is photolysis. When the light reaches the light harvesting complex here, it is connected with what? Splitting of water. So water is splitting. Look at that. H2O is electron is produced and then hydrogen, 2 hydrogen, 2 H plus is released and O2. Here oxygen is released. So the first product of photosynthesis, sometimes they may ask, what is the first product of light reaction? We may say product of light reaction means always we remember ATP and NADPH only we remember. No, first you should remember oxygen. Oxygen is the product of first light reaction first. Okay, here by splitting of water, oxygen is released. That's very, very important. And the electron enters to the system and then the system becomes what? Excited. From normal state, ground state to excited state it goes. Okay, so the electron transfer occurs here. Electron is transported now. Okay, because two important processes connect. One is light reaches the reaction center and because of that photolysis occurs here. What is released? Oxygen is released and the electron is transferred to the system in the chain of reaction. So that's the first thing. Okay, now the electron is actually transferred through the electron transport chains here. See, later we will be studying many chemical molecules. Here also you can see, here I have mentioned so many chemical substances basically in the electron transport chain. So here many substances come. So that okay, to the electron transport chain, the electrons are transferred. Okay, electrons are transferred. And then look at that, the light. These electrons also reaches the light harvesting complex. Here only, do you remember? P700, P680. That also you should remember. Okay, so now photosystem 2, 1. So again, this part we saw. This is what the cyclic reaction part we saw. Okay, cyclic reaction part there we saw basically. Okay, cyclic reaction part. So from the Z scheme, these two light received and then to the electron transport chain. Here the photosystem 1 again receives the electron. Now electron acceptor. The other photosystem one substances, molecules, different chemical substances we saw, they comes as what? Electron acceptor. And then what is formed? Again, because of the electron, NADPH is reduced into NADPH. Okay, NAD plus to NADPH. So this is the Z scheme you can see here. So light comes here, photosystem 2, and then electron transport chain, and then photosystem 1. Okay, so you can see this scheme, they basically call it as what? Z scheme. Here, the main thing you should understand is, it is, that's why, see, look at that, it, there is, comes, goes like this, comes, goes like this. So, no cycle of phosphorylation. Phosphorylation means addition of phosphate group. Look at that, this addition of phosphate group to produce ATP. Okay, to produce ATP. To NADPH, phosphorylation, NADPH plus. So, it is non-cyclic. For that scheme represents the non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation. Okay, fine. So this is very, very important. And another important thing you should understand is what substances can inhibit in the non-cyclic photophosphorylation or Z scheme. Some substances can inhibit that one. So remember these substances: 3,4 chlorophenyl 1. Okay, 2, 3, 4 chlorophenyl 1 dimethyl urea. Okay, so these are all the substances. They call it as what? CMU, DCMU. At least remember the abbreviation of the chemical substances. Okay, here little spelling mistake is there. It is UREA, no S comes. Okay, and then 3, 3, 4 dichlorophenyl 1,1 dimethyl urea. Okay, CMU and DCMU are the substances. In one mark they may ask, CMU and DCMU are the substances which inhibits that scheme. Okay, so that scheme is called non-cyclic photophosphorylation, and then the previous one which we saw is cyclic. This is cyclic photophosphorylation. So they are the components of what? The light reaction. Now look at the products of light reaction. ATP formed. Okay, ATP formed here. One ATP in cyclic photophosphorylation, ATP. Okay, so 2 ATPs are actually formed. Okay, and then look at that, NADPH formed. Okay, so this is very, very important. And then oxygen formed, the products of photosynthesis. And now we'll finish just in 2 minutes, another 2 minutes. So chemi-osmotic synthesis phosphate. So look at that, just the system alone, the diagram alone, let me explain. Please understand this diagram. Okay, so the photosystem 1, see, look at the electron transport chain occurs. So this is the membrane of the thylakoid. In thylakoid membrane only, look at that, photosystem 1, and then the electron transport chain complexes, and then photosystem 2, photosystem 1, and then it occurs. See, this is what we saw there in Z scheme. Okay, so like this, the electron transport chain occurs. Meanwhile, the products, look at that, NADPH, and here the ATP. So that substances are formed. Okay, ATP and NADPH, that substances are basically formed. Okay, in chemi-osmotic synthesis, see, H plus, look at that, H plus, this H plus, and then even in splitting of water, look at that here. Here also H plus are formed. So the proton concentration is increased, and the proton concentration, the protons which are produced, in 2 minutes I'll finish, don't worry. The protons which are produced accumulated in the lumen of the thylakoid. See, in the thylakoid membrane, electron transport chain occurs, but in the lumen, inside the thylakoid membrane, here, look at that, the gradient increase, the concentration of H plus ions increases. Okay, so electron is transported, the negatively charged electron is transported along the membrane, and the protons which are formed are accumulated. inside the membrane. Okay, so there is increase in concentration of H plus ions. This makes, look at that, in the thylakoid membrane, there is an enzyme pump. There is a pump. This pump is called actually what? Proton pump. Okay, H plus ion, proton pump is there. So that proton pump, look at that, it has two parts. Transmembrane, F4 portion, and then out of the membrane. Okay, outside the membrane. In one portion is embedded inside the membrane, F4 portion, and then F1 portion is outside. Yes, ma'am, finish 1 second. Okay, so because of the pump which is present here, ADP, this is ATP pump. Yeah. Yeah, thank you. ATP, ADP, because of the pump, ADP is transported into ATP. So here also ATP is produced, but no, here because of electron transport chain, and here it is because of what? The proton pump concentration. So this is chemi-osmotic synthesis. Thank you. Take care. Question part we will discuss. This is very technical part. Question part we will discuss later. Thank you. Thank you.
Thank you, sir.
Thank you. Thank you. Sorry, today we're not able to discuss, but later we will definitely discuss about all these things. Thank you. நாளைக்கு ஒர்க் ஷாப் இல்லை தானே சார்? ஃபர்ஸ்ட்